Dr. Lisa can be ready within 4 weeks, 1 week's time. Just look for my
Start countdown now.
to people check the support for the teaching.
Okay, right.
Ready.
Just tell me when you start. Right. Second, I know. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Salam Ramadan and good evening to all. My name is Dr. Noliza Abdulatif. On behalf of Innovation Center in Agritechnology for Advanced Bioprocessing, ICA, and also ICA Director, Professor Siang at Universiti, I would like to welcome to all audience to ICA webinar series number 7 with the theme of bioeconomy, food security, and healthy life. Today, we have speakers from our neighbor country. So Yvonne. Hi, Dr. Yvonne. How are you?
Hello, everyone.
Okay, thank you, Dr. Yvonne, for joining us. Okay, let me introduce about Dr. Yvonne. Dr. Yvonne is currently a research fellow at Singapore Center for Environmental Life Science, Teknologi, with a top research center located at Nanyang, Universiti, Singapore. She has been joined this institute since 2021. She received her PhD in biochemical engineering from Putin University, Malaysia. She has been experienced in biotechnology, focusing on single cell protein and polar carbohydrate production using methanotropes, which is a bacteria that convert methane to valuable material. Her work contributes to climate change mitigation by transforming waste methane into eco-friendly high-value products to support circular bioeconomy research. To ensure a smooth and excellent session, we also have Dr. Zaidah. Dr. Zaidah is a senior lecturer at the Faculty of Science, and he also holds a position as manager of Clinical Management Unit at the Laboratory Management Center. He also our research fellow. Dr. Zaidah earned a PhD in plant molecular biology and botany in Imperial College London as a dedicated plant biotechnology. He has been an active member of the Croatian Botanic Society since 2019. Her research focuses on plant biotechnology, particularly the study of plant protein for various applications. Her work spans from protein profiling of local Malaysia, medicinal plants, to in silico molecular modeling and downstream product development. Okay, for today's session, the title is Scaling Methane from Gas Waste to Sustainable Product. I am sure everyone will enjoy this webinar. Once again, thank you to Dr. Yvonne for accepting our invitation to be our guest speaker, and also Zaidah for moderating this session. Thank you again, and let's make this webinar a useful and inspiring experience. Thank you. Over to you, Dr. Zaidah.
Okay, thank you, Dr. Nolila. Assalamualaikum and good afternoon, everyone. So I hope everyone is relaxing and hopefully we will enjoy Dr. Yvonne's talk in a while. Let me introduce Dr. Yvonne to you. In extension to what Dr. Nolila has said about our speaker, Dr. Yvonne, he actually specializes in microbial biotechnology. Her focus is actually on single-cell protein, SCP, if anyone is familiar with that, and also polyhydroxybutane or PHP. This is by using methanotropes, or bacteria that convert methane into valuable biomaterials. So her research actually explores their growth under atmospheric and high pressure conditions and optimizing their potential for sustainable food production alongside biodegradable products. So her work basically contributes to climate change mitigation, and this is by transforming waste methane into eco-friendly, high-value products and also supporting a circular bioeconomy. So I'm sure without further delay, I would hand over the floor to Dr. Yvonne to basically go ahead with the talk. Over to you, Dr. Yvonne.
Okay, thanks. Okay, I'll be sharing my slides. Wait ah. Okay, so first of all, I'd like to thank ICA for inviting me today to be the guest speaker. It's my honor to be able to be here to share my project. So today I'll be going through a topic on upcycling methane from waste gas to sustainable products. So before I go into the details, I'll just give a brief overview. So today I will be sharing on how methane, a potent greenhouse gas, can be transformed into valuable products like protein-rich food and also biodegradable plastics using a special bacteria called the methanotrophs. This process supports the bioeconomy, it enhances the food security, and also it provides a sustainable solution. And my presentation today will cover what methane is and why it matters, and how do methanotrophs work, and how do they produce sustainable byproducts, and some of the economic and environmental benefits, and also key points and challenges. So before I start my presentation, I would like to introduce you to CELSI. So CELSI is a Singapore Center for Environmental Life Science Engineering. It's an interdisciplinary research center of excellence, RCE, which is funded by a few key institutions in Singapore. So it's funded by NRF, the National Research Foundation, the MOE, Singapore Ministry of Education, NTU, Nanyang Technological University, and also NUS, the National University of Singapore. So CELSI is hosted by NTU and NUS. So if you are interested with what CELSI is doing, you can visit our website here, www.celsi.sg. So our research focus area is on biofilm and microbiomes. So this is the major area. And some of the team research includes also environmental engineering, the metaomics, and also public health and medicine. So in order to support all this research, we do have some state-of-art facilities, which includes the advanced sequencing technologies, the imaging facility, and also we have a range of bioreactors from laboratory to full scale. So we do have services for sequencing and also for imaging, and expertise in setup of bioreactors. So if you are interested, you can also reach out to us. Okay, we do also provide this graduate program, PhD program. So for local university, which is Singapore, you need a bachelor degree with at least a second upper. And then from overseas, so in this case in Malaysia, so you need a bachelor degree in first class honor. So some of the other considerations that will be taken into consideration, it includes the research proposal, research and work experience, awards received, and also the number of the papers published. So the research covers the full tuition fee, monthly stipends, IT allowance, and also conference allowance. So every year the admission intake is in August and also January. And for more information, you can email to this CELSI-graduate program at ntu.edu.sg, and this is the website. So currently there's two positions that is open. It's under Prof. Cynthia and also Prof. Laura. And I think the closing or the deadline is on 30th of March this month. So if you are really interested, you can take a look, and then just search up this website. You can find the advertisement in this. So okay, today let's start our topic. So how and why do we have to create a sustainable food future by 2050? According to this World Research Institute, in 2050, the number of population is estimated to increase to 10 billion. So in order to feed this amount of people, we do need about 56% more food So how do we feed 10 billion of people without using more land? So we are currently using 50% of the world's vegetated land for agriculture. So further land conversion will only increase carbon emissions and also increase the biodiversity loss, the deforestation. So we do need some innovative that's be able to allow us to feed 10 billion people without using more land. And also, how do we do this while lowering the emissions? So in 2010 alone, there's actually 12 gigaton of carbon dioxide equivalent. So how do we achieve this? How do we lower these emissions by 2050 by 67%? We can do this with innovative technology, which includes improved feeds, plant-based burgers, resilient crop beats. But then in today's talk, we'll be addressing this emissions problem and then how do we feed 10 billion and then without using more land. By using single cell protein, we can use the greenhouse gases and then turn it into food to feed people. So methane itself is a problem and also is also an opportunity. Methane is a greenhouse gases that's 25 times more potent than carbon dioxide, which contributes to global warming. And then when you see the heat map here, you can see that some of the country actually has a very high amount of methane emissions. It reaches about 1 billion ton. So the methane emission itself is a global issue. So, but then where does this methane come from? First, it comes from waste, the anaerobic digestions. It comes from landfills, wastewater treatment, and also food waste. Next, it also comes from fugitive emissions from the oil and gas industry, and also from agriculture, from rice plantation, and also from livestock. So all these industries, all these waste, right, they actually serve as a very good carbon source for us to turn it into a valuable product. So this is to achieve a circular economy. Circular economy. So how do we achieve this? We can use a type of bacteria, which is called methanotropes. So these methanotropes actually uses methane, and when it combines with oxygens, it produces methanol. So this process this process uses enzymes such as SMMO and also PMMO. And SMMO is iron dependent and PMMO is copper dependent. So without iron and without copper, right, this enzyme doesn't work. So it will stop the conversion of methane to methanol. So it's very important to have iron and copper in the growth medium. And then from here, we have this methanol. Methanol, then it will be converted to formaldehyde, and then it will be assimilated to biomass. So to assimilate it into biomass, it has different, it undergoes two different pathways, which is the IUMP pathway and also the serum pathway. And this pathway, it really depends on what type of methanotrope there is, what type of methanotropes. If it's type 1, then it will be using the IUMP pathway. And if it's type 2, then it will be using serum pathway. And if it's type X, then it will be both. So this is an example of how methane valorization works. So we have these waste streams, and then through fermentation, we produce it into consumer product. So here, the first pictures here, you can see that from waste management or any other activities that producing methane, we can actually direct this methane into a fermentation here. And then here it will be converted into biomass. So this biomass can be harvested, and what we can separate it by using centrifugation. And biomass, it can be further downstream processed into SCP, PHP, PHS, or EPS and biofuels. And then the liquid fraction, then we can undergo a series of reaction, hyperosmotic shock, cation exchange, and then evaporation to obtain this pure ectoin. So this byproduct itself is actually very useful for other consumer products. It can be used in cosmetic, it can be used in medicine, It can be used in pharmaceuticals, vitamins, bioplastic also, and also food production. So like I've mentioned earlier, we can address the greenhouse gas, the climate change issue, and also the increase in global food demand by using alternative proteins. So alternative proteins is it's categorized to different. There's few categorization. First is the plant-based protein, lab-grown meat, insect protein, or the single-cell protein. So today we only cover single-cell protein. Single-cell protein can be produced using bacteria, fungi, algae, and then it's actually a dried form of microbes or biomass. There's or protein that is extracted from these microbes. So what are the advantages of bacteria SCP? So some of the nutritional aspect includes the high protein level. So it's greater than 65%. Sometimes is if the bacteria can produce higher quality, it can produce up to 80% of protein. And then it also has all the essential amino acids. It's rich in vitamins B3, B12, and also it's also high in fiber. And bacteria itself, it has high growth rate, and also it has flexibility in the use of different substrate. It can be used in, it can use carbon dioxide, it can use methane, hydrogen, oxygen, glucose, and etc. And also its independence of cultivable land. So here we can achieve the production of proteins without using more land. And also we can address the climate issues. And then it's also a year-round production. So SCP itself, right, the single-cell protein, bacteria single-cell protein, it has been extensively used in aquaculture. So these are some of the examples from feeding trials. So there are few companies that is producing single-cell protein. So we have this Calista, Unibio, and String Bio. They are all producing single-cell protein, which is FitKind, Uniprotein, and Prodigy, by using methanotrope strain named Methyllococcus capsulatus. So all these strains, these single-cell protein, researchers use it to replace soy meal, fish meal in aquaculture feed. So this feed, they use it in different aquaculture. They use it in sea bass, juvenile giant carp, the Pacific white shrimp, and also rainbow trout. And the result shows that it's actually benefiting their growth performance, improving digestive enzymes, and improving the immune parameters. So it also improves the disease resistance and has a higher weight gain. And also it reduces feed conversion ratio. So here is a figure of how much of carbon that's emitted by producing 1 ton of soybean meal, single-cell protein, and fish meal. So you can see here when the production of microbial single-cell protein of 1 ton, it actually has a negative carbon emissions, whereby production of 1 ton of soybean meal and also fish meal, it produces 7,000 and also 3,000 of carbon dioxide. So here we can deduce that, or we can say that SCP plays an important role in replacing fish meal and soybean meal. It decreases the reliance on marine ecosystem, and also it reduces carbon emissions, and it lowers the cost for aquaculture feed because it reduces the feed conversion. So means that there's less feed that is required to obtain similar weight gain, and also it is increase the yield of aquaculture, and therefore it is able to meet the growing global demand for foods. Okay, so here is another example. This is an anaerobic digester. So here in anaerobic digestion process, it actually produces biogas. So biogas consists of methane and carbon dioxide. And this biogas itself is a great carbon source for single-cell protein production. So here we have economics between the electricity generation, and also the SCP production. So the same amount of biogas and digestate, if it's used to produce electricity and also single cell protein, you can see that single cell protein pathway actually produces a greater net profit than electricity. This is because single cell protein itself has a higher market value than electricity, and SCP can be used in a lot of industry. It can be used for human consumption. It can be used in aquaculture and also even animal feed. But like previously discussed, SCP is widely used in aquaculture, but what hinders the SCP or the microbial food from reaching our plants? There's actually a lot of factors, but today I will only discuss a few. The first is the regulatory aspects. So the safety of microbial foods is actually overseen by a few agencies. So we have EFSA, we have FDA, Singapore Food Agency. So these agencies, they have to ensure that the microbial foods is safe for human consumption. And especially if it's a new species or it's an engineered species, they are stricter. And SCP, it has to be safe for consumption. So the final product, the SCP, it should not contain more than 2% of nucleic acid. First, when you have a nucleic acid when you consume nucleic acids, if it's break down in the body, it will turn into purine. And then once it's further break down, it will turn into uric acid. So this, if you have high amount of uric acid that's in your body, it will actually cause gout and also kidney damage and cardiovascular disease. So this is why we doesn't want SCP to have higher than 2% of nucleic acid. And the third one is the technical advances. So to produce large amount of SCP, even though it doesn't need land, it does need a high capacity of fermentation technology. So when the conversion or the production of SCP is a aerobic process. So aerobic process, it requires oxygen transfer. So when there's a high oxygen transfer, there's a high respiration. So when you have high respiration, it produces heat. You need an efficient cooling system. And the last one is customer acceptance. So for SCP to be served as a human food, the most important one is customer acceptance. The SCP itself has to taste good. The texture has to be good as well. And of course, the price has to be cheap or affordable with high nutritional value. So currently, there's not much of microbial protein that's in the market. So the first one, the first microbial foods that's launched in 2023 is called the Solen, produced by Solar Foods. So it's approved by SFA in Singapore in 2022. So in order to have customers to accept the use of SCP as food, they actually have this food testing in May 2023. So this microbial protein is made from chemoautotropic bacteria. They only use air and electricity in the production. So the main ingredients they capture from air includes the carbon, hydrogen, oxygen, and nitrogen. So these are some of the examples that they use to the SCP that they produce. So they use it as an alternative for meatballs, also omelette and ice cream and protein bar. So this is the first microbial foods. And next is corn. This you guys might be familiar. It's quite widely sold in supermarket or hypermarket. It's actually a type of meat replacement from fungus called the Fusarium vanillinatum. So this fungus is actually found in soil. So they have this fungus in a fermenter. They grow it under glucose and other nutrients, and also they supply it with the necessary trace elements. Then once they have this biomass, they further process it, they reduce the amount of nucleic acid to below 2%, and then they texturize it with egg to create this meat replacement here. So like in previous slide, we have mentioned that methanotrophs can produce SCP. So it can also produce polyhydroxy butyrate, PHB. So this is a form of bioplastic. So it's similar to synthetic polymer like the polypropylene. PHB, it can be is produced by bacteria, and then is in the forms of granule that's in the cells here. And then it's a form of storage material, and it forms under excess carbon and energy, but then it has to be under limited nutrients condition, such as limited nitrogen, limited phosphorus, or oxygen. So this PHB can be used in agriculture for coating for controlled release of fertilizer. It can be used for tissue engineering applications. It can be used as a packaging for biodegradable plastics. It can also be used in the pharmacological, as in the drug delivery system. So this PHB research is widely researched by other people. So some of the company includes Cardia Bioplastics, Kaneka. So here you can see that they actually produce it they use this PHB for the production of plastics, cups, bowls, and also forks or utensils. And here, besides Cardia and also Kaneka, we have other companies as well, Like Technologies, Yvonne, Bioon, Denimo, Scientific, and etc. So there are a lot of factors that's actually affecting the cultivation of methanotrophs. So I will only go into a few today. So the main one is actually carbon. So methane plays an important role as a carbon source for methanotrophs. But the issue with using methane is that it has very low solubility in water. So it's also important to have methane oxygen optimized methane oxygen ratio. Here we can see a stoichiometry equations for one of the species, the Methylococcus capsulatus, that's widely used in SCP production. Here you can see that 1 mole of methane is actually using 1.5 mole of oxygen for the production of biomass. So it's not always 1 mole to 1 mole, and it's dependent of the type of strands, and different species has different requirement for oxygen. So this is one of the factors when we use methanotrophs for cultivation into valuable byproducts. The next one is a reactor design. So like here I've mentioned, methane has low solubility, right? So it's important to have a reactor that can address this gas liquid mass transfer. So Calista, Unibio, and String Bio, the production of SCP, the large scale production, they are actually using this U-loop fermenter. So here you can see from this U-loop fermenter, they have the biomass in this U-loop fermenter. They have this methane, nitrogen, and oxygen that's recycled and circulated in this U-loop fermenter. And when these are when this methane runs out, or any nutrients runs out, they will replenish it in this U-loop fermenter. And finally, when there's a the biomass collection is at the end of the reactor, and they can collect it and then harvest it and then separate it for further process. So U-loop fermenter is just one of the reactor that can be used for methanotroph cultivation. So I've also studied high pressure reactor by using high pressure to increase the solubility of methane in our studies. And other than high pressure, we can also explore the membrane bioreactor. It also serves as one of the solution to increase the gas liquid mass transfer. Next is also nitrogen. The nitrogen forms and also concentration. So nitrogen, it presents in the form of ammonium and nitrate. So some of the species or some of the strains, they actually prefers ammonium than nitrate. So ammonium can be directly used for amino acid synthesis, but then nitrate needs to be reduced. But despite the hassle here, we do have some methanotrope strain that is prefer nitrate over ammonium. So this is also another important composition that has to be optimized, and also the concentration is also quite important. So it depends on the applications. So for SCP production, the concentration of nitrogen it should be high enough to support the protein synthesis. But in the case of PHP production, it needs to be low enough to create this nutrient limitation condition for PHP to be formed. Last one is the strength. So like I say, every different species or every different strains of methanotropes, it requires different carbon, nitrogen concentration. So it's always essential to select strains with higher growth rate. And these strains is always affected by different nutrient composition or growth conditions. And like if the application is for the production of SCP, so of course we have to choose a strain that has a high growth rate and maybe produce high essential amino acids or higher protein content. But if you want it to produce PHP, then we might need strains that can survive or produce biomass under low nitrogen concentration and then has a high production of PHP. So it really depends on what type of application, and this is always strains and species based. Okay, so today I'll just briefly go through what I've done in my research for these past 3 years. Some of the results isn't really published, so I can't go into detail. So I'll just briefly go through what I have done. So initially, we started with enrichment of methanotropes from different natural sources. So our first step, we use this metagenomics mining for methanotropes gene. We have the bioresource libraries from different environmental samples, and we mine for their gene, PMOA and MMOX genes. And once we have these positive hits, we'll proceed with the collection of the sediment. Then once we have collected the sediment, we'll incubate it in a serum bottle at 30 degrees Celsius, and then we will subculture it a few times until we have a stable culture. So here we can monitor their growth by using GC and also UV visible spectrophometer. So GC is to monitor the methane consumption, and this UVVis is to check for the turbidity. So once we have a decrease in methane, which means that there's the bacteria in this serum bottle is actually using methane, and there's an increase in turbidity. It also proves that there's a growth of methanotropes in this serum bottle. So once we have this stable microcosm, we plate it on the agar plates, and then we incubate it in these offered jars with methane in it. So we'll continue shrink it until we have these pure colonies in it. Then once we have these pure colonies, we check their purity using this phase contrast microscope and some non-specific media such as TSA, the LB or NA, nutrient agar, to check whether there's any growth. If there's no growth, it means that the bacteria, the strains that we have, it should be pure. Then we subject it to DNA sequencing. So these pure strains, it will be subjected to further optimization. So these are just some of the results that we have it here. So this is the microcosm that we obtained. This is slightly pink in color. And then we have this, and then we spread it on agar. Then we can see there's actually different types of colonies in it. So when we select, and then we streak it at agars until we have pure strands. And here we have 3 different types. So the first type is a strand UP105. So this is white in color or cream in color, and the size is actually larger than other strands. And here, if you see that there's this white granule, this is actually PHB. So strand UP105, they can produce PHB. And then for strand UP106 and this UP301, it's actually orange and pink in color if you can see from the pictures here. So these are the pigmentation for these are the pigmentation. So this pigmentation is because of the production of carotenoid. So this carotenoid is a source of antioxidant and also vitamin. So which means that we have 1 strand that's able to produce PHB. We have 2 strands here that's able to produce vitamin. But all these 3, it can be used to produce SCP. So it really depends which strand has a higher growth rate and better protein content. So we took one of it, which is a UP105 that can produce PHB and biomass, and then we studied it under different nutrient composition. So here I only showed you the nitrogen, copper, and iron, but in fact we did study the methane to oxygen ratio and other compositions, the temperature, the spinning, the stirring speed, and etc. But that doesn't really affect the biomass and PHB production. So here we finalize it. I mean, we streamline down. There's these 3, copper, nitrogen, and iron, that's actually affecting the PHB production. So if you see it carefully from this figure, you can see that copper, nitrogen, and iron, it doesn't really affect the biomass production. It's more or less the same, about 0.7, 0.6 gram per liter, but then it is affecting the PHB production. So here, when we have lower nitrogen, which is ammonium in this case, it actually produces higher PHB. So this is because it has a limited nitrogen, which triggers the production of PHB. And then for copper and iron, this is affecting the enzyme that's that is used for the conversion of iron methane to methanol. So this is also important, the enzyme, as it's a core factor for it to work properly. So we can see that a higher copper, it doesn't really help with the PHB production. So a lower one will do. But then for iron, it does need a higher iron. But then even higher, it will inhibit the PHB production. So therefore, it's important to have optimization on different nutrient composition. And if we see from just this result here, you can see that the biomass production is actually about more or less 0.6 to 0.7 or 0.8 gram per liter. It depends on which nutrients. So for PHB, it's about 0.4 to 0.5 gram per liter. So we also established a method called the lipid green 2 PHB staining. So initially, when we tried to identify this, to quantify this PHB in our cells, we use now red and now blue. But then these 2 dyes, right, it doesn't really give us a very trustable result. It overestimates the amount of PHB that's in the cells because it stands to the cell membrane. So then after a trial and error, then we found out that this lipid green 2, which is a pretty recent fluorescence dye, and then it gives a very pretty accurate result when we test when we stand it with cells and PBS, and then we subject it to spectrophlorometer under excitation 440 nanometer, emission of 520 nanometer, we can stand this PHP quite nicely, and under micrograph we can see it. We can see the PHP granules here. So if we compare this method with the conventional method, the conventional method includes the extraction of these cells, the PHP here, and then we subject it to solvent extraction and GC to identify the amount. So often that the conventional method, right, it requires longer time. It requires more chemicals in it. It also requires more volume. So for example, to identify the amount of PHP that's in a cell, we might need a 1 ml to 5 ml of cells to extract to have a sufficient amount of PHP to be quantified. But then in this case, when we use spectrophlorometer, we don't really need much of the cells. We can just use 0.1 ml that's in the serum bottle or in the batch reactor. We took it out and then we add it with lipid green, and then we can just plug it and send it into the spectrophlorometer, then we can get the results. So therefore, lipid green 2 is also a fluorescence that provides quick and effective method for PHP detection during fermentation, as this lipid green selectively stands PHP and it has minimal interference from cell membranes. So this is one of the research paper or review paper that I found published in 2024. So this is their summary on what others found. So in their batch system, they actually have biomass yield of 0.49 to 0.585 gram per liter. And in our case, in our batch mode as well, we have our strand that's producing 0.6 to 0.8 gram per liter of single cell protein. So can we say that it's better than the existing strands or the strands that the other researchers found? We can't really say so, but we need further research to validate this hypothesis. Yeah, so comes to our summary. And methane is actually a waste to value opportunity. It can really be used in the production of many byproducts, not just SCP and PHP. We can turn it into a ectoin for cosmetic, vitamins, which has a probably better market values. Methanol can be used for protein and bioplastic industry, and it addresses challenges such as land constraints and also climate change. But this cultivation of methanotropes to bioproducts still has a lot of challenges. First is the gas liquid transfer efficiency. So methane is poorly soluble in water. So this can be addressed with reactor design, but then reactor design is always a challenge. The second one is scaling up of the production will maintain low production costs. So just an example of purification of PHP from cells. This step itself is still a very difficult steps that requires a lot of cost. The next one is the regulatory approvals for food and feed use, and also the public acceptance of SCP as food. So some of the future directions, but it's not limited to, but there's a lot, just two of it that I'll be mentioning today. So first one is reactor design. So it's important to have a reactor designs that maximize this gas liquid transfer efficiency. So in our case, in my study, I focus on the use of high pressure bioreactor. And for the second one is using waste stream effluent as growth medium for nutrient recovery. So we can use pure methane, we can use biogas as a carbon source. We can also look into different waste stream. We can consider using the digestate or the effluent that's in anaerobic digester to be growth medium. Or another example is if it's in Malaysia, there's a lot of pome, the palm oil meal effluent. We can also use this as a growth medium, so for further nutrient recovery. So there's actually a lot of waste stream that can be considered as growth medium. So I would like to thank the financial support that's provided by A-Star, Singapore Food Story, and also CLC Seed Funding in supporting this whole project. And I would like to thank my team members, Dr. Stephen, Dr. Te, Dr. Muhammad Fidaus, and Dr. Jamie Hings and Prof. Sanjay for their support. And if you have any inquiries or if you have any ideas that you want to collaborate, you can email to me, xiang.he@ntu.edu.sg. So thank you for your attention. And now we'll go into question and answer.
Okay, thank you, Dr. Yvonne, for that very interesting and refreshing talk on your work and also what basically could be done to the methane, because as we know, methane is actually a big problem everywhere, isn't it?
Yeah.
Right, so from our Facebook, we have 3 questions, and I see that we have like around 79 to 80 audience at the moment. So the first question is, how can upcycled methane be safely and effectively integrated into food production? And what are the key regulatory challenges in commercializing these food products? This is a question from Yiik from Facebook.
So how can we upcycle methane be safely and effective integrated into food production? What are the key? Yeah, so like I've mentioned just now. So what hinders these microbial foods to be for human consumption? It's always hard because if you are the consumers and I tell you that this single cell protein is from bacteria and we get this bacteria from sediments, do you accept it? It's quite hard, but we do need more awareness in letting consumer knows that SCP is it has a lot of nutrition, is high in nutritional, and key regulatory challenges in commercializing this food. So it's like I said, it's the FSA, SFA, EDSA. It's not easy to be commercialized. You need a lot of human trials. You need a lot of laws and regulation to be approved. So now we only have currently we only have one, which is Solian, produced by Solar Foods in this microbial protein. So I don't have any specific answer, but then it's just hard at this moment.
Okay, all right. So I think it is almost similar to the second question because I'm not sure if I'm basically saying the name correctly. Deepash Nisivakumar. Okay, what safety regulations and testing processes are involved in approving microbial proteins for human consumption?
Okay, this is very specific. Sorry, I can't answer you because I'm not involved in the regulatory agency. But I think one of it, like I discussed earlier, is the nucleic acid content. So it has to be low enough that it doesn't cause any health problem. And most probably that's one of it. And then if it's engineered species, right, it has to be safe and then it doesn't affect our DNA. So it needs a lot of clinical trials to be approved. So specific answer, I can't really answer. If you want to know, you can check I think SFA Singapore Food Agency actually have these full requirements on what are the safety regulation and how do these microbial protein can be consumed by human consumption.
Okay, all right. So our third and final question. Well, from the audience, I also have one question or two, okay, for you. So the question is, are there specific waste streams, such as agricultural and industrial methane emissions, that are more suitable for methanol trough-based fermentation?
Okay, so waste stream. So we actually use biogas from anaerobic digester, and it does produce. I mean, it's suitable to be used as a single cell protein production. And we do have one FRP student that's in UTM right now that's using POME, palm oil meal effluent, as one of the liquid medium. But we don't have the results yet. So if there's anything, you will see it in the publication.
Okay.
Yeah.
Right. So I take it that Dr. Muhammad Firdaus is the one from UTM, isn't it?
Yes.
Okay, because he is in my department actually.
Yeah, yes.
Okay, Dr. Yvonne, okay, those are the questions from Facebook. I actually have, okay, 1 or 2 questions for you, okay, based on your presentation. Because I'm a protein person, so I'm working on basically plant-based protein, but I'm actually also interested in what you have presented just now. And my question is basically on methane itself. So what are your thoughts, okay, on the methane produced from landfill production and also from compost?
Okay, so if it's methane from landfills and also from compost, is when you send it to fermentation and it's used by the bacteria itself, it's already a cleaner approach. If you compare with directly, you are using waste streams from, I mean if you are using directly, but if it's a term of if it's a gas that is from compost and landfills, it's a cleaner approach.
Okay, so the
We
landfill is a cleaner approach because they have a mix of, well, waste, isn't it?
But for methanol troughs, we are actually using just gas, right? We don't really use the compost and the landfills. I mean, yeah, the
Okay.
waste. We are extracting the methane itself from the landfills. So if we pass it through a filter and then through the fermenter, then we can safely produce SCP from it.
Okay.
I think that's my thought. Do you have any thoughts in it?
Okay, understood. Okay, and that is actually something very interesting to ponder on because I know a few of us, okay, over here in UTM, which is also working on compost. And because I visited a landfill area, okay, somewhere in Selangor last year, so it was interesting to see how they actually convert all those gases produced.
Mm.
All right. So
I
my
think
last question.
the common practice right now, they are using the biogas or the methane emissions for electricity, most of it. Yeah, to power up your own plant. And if you want to use it for SCP production, it should be another sustainable solutions. We can look into it as well.
Okay, all right, interesting. And my last question to you, have you ever tried the foods that you mentioned just now?
No, I didn't get to. Yeah, I didn't get to. Just I mean the food testing is in Singapore. But then when I realized, right, it's already past time. Yeah, so yeah, if I realize, I would have tried.
So is it only available in Singapore?
Currently, yes, because
Okay.
it's only accepted in Singapore. So yeah.
Okay, all right. So thank you very much, Dr. Yvonne. So basically what I could conclude, okay, from your talk just now, very lengthy but also very interesting talk, okay, is that we actually have ample production of gases and it depends on where they are being produced. So your work, okay, aiming or focusing okay, to basically recycle those into valuable products, especially by using SCP and also you're using PHP with it. So I would say that, okay, those are very promising area. Okay, and in the beginning, you also have one slide okay, showing that you accepted students. Okay, that means that it is from not only from Singapore but also from Malaysia or from anywhere in the world, isn't it?
Which one?
In your earlier slides, okay, there is a mention on student internship, or is it a work?
You mean graduate program?
Yes. Oh, yeah, graduate program. Okay, all right.
Yeah, PhD. Yes. Yeah, okay, we are accepting students from overseas as well. So if there's any students here that's listening, if you want to continue your work in biofilms or micros environmental engineering, feel free to submit your ideas to us. And then there's 2 opening right now
Okay, all
under
right.
Prof. Cynthia and Prof. Laura. So you only left another 2 weeks to apply. So if you want to apply, you better hurry up.
Okay, all right. So if there is any student here, okay, in Facebook, so this is your opportunity, okay, to
Yes.
apply. Okay, you can either contact Dr. Yvonne or basically contact ICA if you didn't get the cash just now.
Yes.
Dr. Yvonne, on behalf of ICA, I would like to thank you for your time, okay, for giving us the talk and basically for providing us into other insights on how to recycle and to make, well, not only the world, okay, but basically everyone in a better living condition. All right, so with that, okay, thank you everyone, especially those who are still online on Facebook. Okay, that is it. Okay, from ICA. So I hand it over to basically Dr. Nolita. Is it? Oh, we are done. Okay, so basically we are done. So thank you for your time again, and we hope to see you again in another series of ICA webinar. Thank you.
Thank you.
Thank you, Dr. Yvonne. Thank you.